This conference will now be recorded.
Assalamu alaikum everyone and welcome. It's 5 o'clock, so we will start today's session. Just before I start and introduce Philippe and Dr. Shazia, I just want to say that please use the chat box only for questions and relevant discussion. Please keep your mics on mute and your web cameras off. I will only be posting the evaluation link in the last 5 minutes of the session, just so that we can make sure that everyone's attention is on what is going on screen rather than what's happening in the chat box. And once you fill out the evaluation form, please give us a day or two to send you the questions. But as I said in the previous session, only people who fill out the evaluation with their correct email address will be receiving those questions, because that is what we will be using. If you miss the evaluation or the session or the quiz or something, to maintain a sense of fairness, we will not be able to send you the quiz or the link immediately after. However, everything that we do is uploaded on our YouTube channel, so you can always take a look there. मैं एक बार उर्दू में भी रिपीट कर दूंगी. प्लीज चैट बॉक्स के अंदर आप अपने रेलेवेंट क्वेश्चंस लिखें, रेलेवेंट डिस्कशंस करें एंड कोशिश करें कि आप क्विज इवैल्यूएशन और सर्टिफिकेट्स की बात ना करें. ये सेशंस आपके लिए होते हैं, आपके क्वेश्चंस आंसर करने के लिए होते हैं. तो अगर आप इसको जितना कॉन्वर्सेशन को रेलेवेंट रखें, उतना सबके लिए बेहतर होगा. अगर एंड में सिर्फ आखिरी 5 मिनट में मैं इवैल्यूएशन लिंक डालूंगी चैट बॉक्स के अंदर, आप प्लीज वो खोलें, अपने करेक्ट ईमेल एड्रेस से उसको फिल इन करें एंड फिर हम आपको उस पर क्विज भेजेंगे. हमें प्लीज एक दिन दें क्विज, क्योंकि हमने बनाना होता है, बाकी स्कोर्स करने होते हैं और फिर सेंड करते हैं. तो एक दिन का हमें टाइम दें. अगर आपको उसमें नहीं मिलता, परेशान मत होएं. अगर आपसे ये सेशन, कोई इवैल्यूएशन या कोई क्विज मिस हो चुकी है, सब कुछ अवेलेबल होगा हमारे यूट्यूब चैनल पे, वेबसाइट यूट्यूब चैनल पे. उसका लिंक भी मैं एंड में डाल दूंगी. और जस्ट टू मेंटेन अ सेंस ऑफ फेयरनेस, everyone has to attend the live sessions, complete the evaluation and complete the quizzes within the given time frame जो हमने दिया होता है to be eligible for a receipt. However, the content will be available to everyone to use as they wish. With that, I would like to introduce Philippe, who I think needs no introduction by this point. Today's topic is on biological risk assessment, a step from the theory to practice, and we'll be going over some case studies, and the Urdu translations will be done by Dr. Shazia Adil again, who is the Islamabad chapter head. Over to you, Philippe. I'm sorry I took so much time.
Yeah, thank you, Mashal. Assalamu alaikum, everybody. Good evening. So indeed, in this webinar, the first section of this webinar, we'll move from theory to practice. At least we start moving from theory to practice. Risk assessment is something that needs to be done a number of times to really be able to do it properly. So that's why I really hope that we'll be able to organize some workshops, some in-presence workshop, where you will be able to work yourself on that. This is not possible right now, so this is why we'll approach the practical aspect through a case study. Before that, I will make a brief reminder of session 2, but I will also give you a few more information. And after the case study, we will see who should really do the risk assessment, and then I will present you briefly a few tools and examples supplied by WHO to help people to really conduct proper biological risk assessment. So during first session, I presented the reasons why WHO wanted to propose a new approach to biological risk management, and really based on risk. And the main reason for that is that they want to give all the countries the possibility to develop proper diagnostic and research capabilities. And for that, they need to be able to implement economically feasible and sustainable biosafety and biosecurity policies and practices. And to be economically feasible and sustainable, they must really be adapted to the situation, taking into account the activity, but also all the capacity that are present in your country. So the risk assessment approach proposed by WHO is actually a 5-step approach. During the first step, we gather information, not just on the biological agent and on the activity, but also on a number of other aspects, including local factors, like what is available locally, what is the capacity of the organization, what are the logistic means, and so on. Based on that information, then we evaluate the risk, and we also evaluate not only the risk on the basis of the biological agent and the activity, but also taking all those aspects into account, like for instance, the capacity of your laboratory, the equipment you have, and so on. On that basis, we develop a risk control strategy. So it's really looking at all the major possibilities and really define how we tackle the biosafety issue in this institution. And then once this is defined, we select and implement a number of risk control measures. So, and then when it's implemented, after some time, we review the risk and risk control measures, either on a regular basis or if there is some major change in the activity, for instance. So this is another representation of that. So we first gather information and evaluate the risk. When we've done that, the last question is that risk acceptable? If yes, we can proceed with the work on the basis of what we have. If the risk doesn't appear acceptable, in that case, we need to find a way to make it acceptable. So we need to develop that risk control strategy, looking at the main aspect that we'll develop, and then select the risk control measures that appear the most adapted to our situation and put them in place, implement them. Once this is done, we should again ask ourselves the question, is this risk acceptable? Are these risks acceptable? If yes, we can proceed with work with those additional measures. If not, in that case, we should really try to look for additional measures. Perhaps there are none, perhaps there are some. If there are some measures, in that case, that you can add to what you already have, and in that case, you should do it and implement, and then it's likely that the risk will become acceptable. There is another possibility, is that given your capacity, local capacity, is that risk is still not acceptable and you cannot do anything just because you are lacking some major equipment or some major capacity with respect to risk control. In that case, one possible issue of the risk assessment should be that we cannot conduct that project. We should not forget that. So the purpose of risk assessment is really to make all activities safe, but in some cases we might just lack the capability to make it safely, and in that case we must also consider the option or not doing that project, at least not now in our current capacity. So what is the purpose of risk control measures? Actually, we do the risk assessment and we define the initial risk, and then we define the strategy and we define a number of risk control measures. The purpose of those risk control measures is to make the residual risk acceptable. So there is a level of acceptability that we need to define. We could say, for instance, we don't want anybody in our facilities to become infected and develop some disease because of their work. And then, if there is some risk, in that case, you put in place a control measure to prevent that people could become sick following an infection, and then the residual risk at that time, that level should be very, very low. So the purpose is really to bring the risk of the activity to a level which appears acceptable. We also saw the risk assessment matrix. That's a tool that we use to visualize the importance of risk and also to be able to prioritize the risk control measures we want to put in place. It's based on the likelihood of exposure. Is it unlikely, possible, likely, highly likely? And then the consequence of an exposure could be very low, moderate, or severe. And depending on that, we could have a very high risk with a highly likely negative event that could lead to severe consequences. That would be very high. And on the other hand, we could have a very, very low risk. A very low risk. And of course, this will help. Of course, we should first act and try to find the right risk control measures for all the risks which are high or very high. And then we can work on medium risk. And perhaps it's less important to work on the low risk if it becomes acceptable, if it's seen as acceptable. Of course, that is a very simple risk assessment matrix. We could use something which is a little bit more elaborate, and that would, in that case, we could have a better definition of the different levels of risk. So back to a kind of schematic risk assessment matrix. According to WHO, most situations correspond to the green area. So most laboratories that conduct biological activities that have a level of risk which is in that green area, meaning that exposure or release would be more or less likely, and also the severity of the consequence would be from weak to moderate. And in that case, WHO is convinced that core requirements, which means mainly basic biosafety practices and with basic protective equipment and quite basic facilities, should be enough to work safely. Then there are some activities that may present a higher likelihood of exposure or release, and also perhaps the consequence would be higher. In that case, you would be in that yellow area, in those yellow areas, and in that case, you're likely to need some selected higher level control measure. I insist on the word selected, because you don't need to put a whole range of control measures. Not like you need to put all the BSL-3 control measures. No, you need to choose what will help you to decrease the level of risk to the green area, or at least to something which is acceptable. Okay? And then there are a number of situations where the likelihood is highly likely. So it's something that is high, that is really likely to present in a number of cases, and also of which the consequences would be very severe for the people or for the environment or the population. In that case, we are in that red area, and in that case, it's likely that we'll require some maximum containment measure. So that means measures which are much more demanding, but in that case, also it's a selection of those measures. We don't necessarily need all types of measures. It needs to be really adapted to the risk. Ooh. Yeah, Shazia, if you could already summarize this this review of last time, please. That's nice.
अस्सलाम वालेकुम जी। आज के इस सेशन में हम नजरिए से अमल की तरफ पहला कदम कैसा लेना है के बारे में बात करेंगे। शूमाकर कहता है कि एक औंस मश्क की कीमत 1 टन नजरिए से भी ज्यादा है, तो इसलिए हम पिछले सेशन की थोड़ी सी याद दहानी करेंगे। खतरे की तशखीस की जिम्मेदारी किसके करनी है उसके बारे में बात करेंगे, और WHO ने एक केस स्टडी भी दी है उसके बारे में बात करेंगे। मजीद WHO ने कौन से औजार और मसाले दिए हैं जिनके साथ हम अपना रिस्क की तशखीस कर सकते हैं। इस चौथे मैनुअल का बुनियादी मकसद ये है कि तमाम ममालिक को मुआशी तौर पर मुमकिना और पायदार बायोसेफ्टी और बायोसिक्योरिटी पॉलिसियों और तरीकों के नाफिज करने के काबिल बनाएं जो इनफरादी सियाको सबाक और तरजीहात से मुताबिकत रखते हों। रिस्क की तशकील WHO 5 मरहलों में करता है। रिस्क के बारे में मालूमात जमा करना, रिस्क की तशखीस करना, रिस्क पर काबू पाने की हिकमत अमली को तैयार करना, रिस्क पर काबू पाने वाले इकदामात को मुंतखब करना और उन पर अमल करना, और 5वां मुंतखब शुदा इकदामात के काबिल अमल होने पर गौर करना। खतरे की तशखीस का अमल और उसके मुमकिना नताइज के बारे में ये खुलासा दिया गया है कि पहला मरहले में मालूमात जमा करते हैं। मालूमात जमा करने के बाद रिस्क की तशखीस की जाती है। जब रिस्क की तशखीस हो जाती है तो हम इब्तदाई काबिल कबूल खतरे को मुंतखब करते हैं। कि हमने देखना ये है कि क्या इब्तदाई काबिल कबूल खतरा हमारे लिए कबूल है तो हम काम को करेंगे। अगर वो हमारे लिए कबूल नहीं है तो हम अपने खतरे को काबू पाने की हिकमत अमली को तैयार करेंगे और मुंतखब शुदा हिकमत अमली को पर अमल करेंगे। इसके बाद हम देखेंगे क्या बकाया खतरा हमारे लिए काबिल कबूल है। अगर बकाया खतरा हमारे लिए काबिल कबूल है तो हम अपने काम को मुकम्मल करेंगे और इसके इकदामात की अफादियत पर नजरसानी करेंगे। अगर बकाया खतरा हमारे लिए अभी भी काबिल कबूल नहीं है तो अजाफी कंट्रोल के इकदामात पर गौर करेंगे और सोचेंगे देखेंगे कि प्रोजेक्ट को खत्म करना है या मजीद नजरसानी करनी है। रिस्क के कंट्रोल के इकदामात का असल मकसद मुंतखब शुदा इब्तदाई खतरे को रिस्क कंट्रोल के इकदामात लागू करते हुए लैब में काम करने के लिए काबिल कबूल खतरा बनाना है। ये 3 इनका रिस्क की तशख़ीस का एक मैट्रिक्स है जिसमें खतरे की अहमियत जानने में मदद देता है, जिसमें नताइज और इम्कानात दोनों को बताया गया है। ये हमारा 5x5 का मैट्रिक्स है जो कि ज्यादा हसास खतरे की तशखीस का टेम्पलेट है, जिसके अंदर नताइज और इम्कानात के बारे में मजीद डिटेल से बताया गया है। रिस्क असेसमेंट मैट्रिक्स और रिस्क कंट्रोल के इकदाम को WHO का ये मैनुअल 3 हिस्सों में तब्दील करता है। वो कहता है कि बुनियादी ज़रूरियात का इस्तेमाल करते हुए बेशतर लेबॉर्ट्री की सरगर्मियां महफूज तरीके से अंजाम दी जा सकती हैं, और ये हमारा सबज हिस्सा है। जबकि कुछ ऐसी इकदाम होते हैं, लेबॉर्ट्री सरगर्मियां होती हैं जिनके लिए हमें मुंतखबशुदा ऊंची कंट्रोल के इकदाम करने पड़ते हैं, जिनको पीले हिस्से में किया जाता है, जबकि लाल हिस्सा उन इकदाम को जाहिर करता है जो कि लेबॉर्ट्री की वो बहुत ही कम ऐसी सरगर्मियां हैं जिनके लिए ज्यादा से ज्यादा काबू पाने के इकदाम की जरूरत होती है। ओवर टू यू फिलिप।
Thank you, Shazia. Yeah, there is one question I think I will answer immediately because it will be short and because I think that it's about the word that is used. Someone says that doesn't understand risk control, and it's probably a matter word. Risk control means reducing the risk or limiting the risk of preventing the risk. And so when we speak about risk control measures, there are different ways to reduce risk or limit risk or prevent risk. And that's what we'll see now in the case study. It doesn't work. Okay. Okay. So this case study would be about rapid blood testing. I took this example from the WHO manual, from the monograph on risk assessment, and I modified it a little bit so that it fits this workshop. I choose this one because I think that everyone is more or less familiar with the kind of issues that we'll deal with, and also because it's a quite simple situation, I would say. So what is the context? Actually, the activity is some rapid diagnostic testing in order to detect blood pathogen, and that's made on blood samples that were collected in the field. And the protocol established by the test manufacturer is that we need to dilute the sample with a specific buffer, and that is done in the tubes that were used for the sampling. After that, we need to do a 5-minute incubation and then mix the sample, and that's done using a vortex. After that, we can introduce the test strips into the tubes, and then again we have a 5-minute incubation time. After that, we can do the reading, and for this experiment, they take a photograph of the sample with the strip with the patient identifier code. Also, the test tubes are still closed and are discarded as such in a closed containers, a closed plastic container, and it's incinerated on site. So it's not always the case like that, but I took this situation because in that case we won't have in this example to deal with waste. So the first step is to gather information. So we need to gather information on a number of aspects. The first one is about the biological agent. In this case, we don't work on a biological agent, but on some biological material, which is blood sample. And then when we look at, when we think of blood sample, there are a number of pathogenic or biological agents that could be present. The main ones are hepatitis B virus, hepatitis C virus, HIV, human immunodeficiency virus, and then some others, but at a lower prevalence, like the syphilis agent and some more pathogens that are normally not present in blood, but during an infection at some time they could be present in blood. So clearly the most likely to be present is hepatitis B virus, and then hepatitis C virus, and then HIV. So after that, we also collect information on the different viruses. And in this case, I put there a number of information about hepatitis B virus. And all those information may be useful to really define the level of hazard in some case. And so, for instance, this HPV is highly transmissible by blood or sexual contact. A very important aspect is that it survives quite a long time outside the host. So if it's spilled, for instance, or on a hard surface, it can survive there for more than 7 days. Also, the forms of the disease, there are different forms, but there is also a risk of long-term complications like liver cirrhosis or primary liver cancer. For that, there is a vaccine that is available. There is also some post-exposure prophylaxis that is available, that is possible. But in this case, it's not available locally. And there is no curative treatment. So if someone gets the infection and we cannot give immediate prophylaxis, in that case, there is no treatment. We can just kind of help the person to feel more or less okay. And same type of information with HCV, for instance. And in that case, there are also acute and chronic forms of disease. It's less likely to survive for a very long time, but there is no vaccine, for instance. HIV, again, the same type of transmission. In this case, the survival outside the host is more limited, but there are also long-term consequences. Somebody who gets infected will have a chronic immunodeficiency. There are still no vaccine available, and prophylaxis exists, but it's quite it's locally expensive. So all those are considerations that we need to take into account. And so the same type of information about the syphilis agent and other agents, we could dig a little bit further, but we probably have enough information there. So and also the other agent could be a very, very different, and the probability, the likelihood that there could be present in blood sample is very low. Anyways, when we look at that whole picture, all these agents are more or less transmissible through blood contact, through cuts, injection, and so on. And but they could also be transmitted through contact with mucosa or non-intact skin. And another aspect I pointed out is that some of them resist for quite a long time when spilled, for instance. So we also need to gather information on the work that will be done. So in this case, it will be done in the sampling tubes themselves, and there will be no transfer of the blood. So there will be no pouring, no pipetting, or anything like that. Of course, this is likely to reduce the risk. Then the sampling tubes themselves are only open twice. The first time to add the buffer, and the second one to introduce the test strip. Then there is a mixing, it's through vortexing, but all that together with incubation is done in closed tubes. So we don't open the tube, we close the tube before doing the vortex, using the vortex. And then the tubes are eliminated as such in plastic containers, and they are incinerated on site. So all those conditions related to the testing procedures, we need to collect them. Then what about the laboratory and the equipment? The laboratory is a single room. It's quite basic, so it's not a fancy lab. It's really basic, but there is enough space. There is no anteroom or airlock, anything like that, and no real ventilation system. But to cool the air, there are a few split units that will cool the air, but it's not ventilating the room. I mean, there is no replacement of the air in the room. The lab is equipped, of course, with benches and a sink, which is interesting for hand washing. There is also a Class 2 biosafety cabinet, but that biosafety cabinet has never been checked and maintained, and it needs checking and maintenance. And also, we think that it will probably need some new HEPA filters. That's not sure, but it's likely. Also, there is no autoclave available for waste decontamination, but as I said, there is an access to hospital waste incinerator, and that's on site, so that would be an easy way to get rid of the waste. So you see, you also collect a number of practical information that are really on the local situation. Of course, that I described here the situation in one lab, it could be totally different in another lab. What about the current biosafety practice? Actually, the lab does not have very fancy activities, and the people are really familiar with basic microbiological practices. with respect to personal protective equipment, PPE, they use a simple cotton laboratory coat, and they also have at their disposal latex gloves. The personnel is there for a long time already. They are trained and they have experience in basic diagnosis practices. Also, they will be trained by the manufacturer on this rapid diagnostic test, and that will be done before the work starts. And also, the few people who work on this project have been vaccinated against hepatitis B. And this, of course, is a very important information. And so the other aspect that we could that we should also consider is, for instance, the fact that the blood samples coming from the patient, we don't have any idea of the health status of those patients. So they could be really ill from hepatitis B or HIV, or they could just be in good health. We don't have any idea. So that means that we need to consider that some of those samples could be contaminated and heavily contaminated. Also, one factor also which is important here is that hepatitis B, HIV, and syphilis are not acceptable in this community because they are associated to sexual practices and so on, and because of that, it's not acceptable, even if there are other. So until now, I've only gathered all the information in view of the risk assessment. Step 2 is really doing that risk assessment, to really evaluate the risk. And so the first step there, the first activity there in risk evaluation is to identify all potential situations in which exposure or release of biological agent. Could you please mute your phone, your microphone? Okay, and so for instance, we use non-breakable plastic tube, and we don't use any needles or real glassware. And that's the impact on that with respect to risk is that there is no risk of accidental inoculation or cuts with contaminated items. So this is a very interesting element. Then we open the tubes. And so when opening the tubes, there is some risk of aerosol generation, and especially after vortexing. After vortexing, you mix the sample, and so you might have droplets everywhere in the tube, but also have a small aerosol. When you open the tube, then the aerosol kind of expands, and someone could be exposed to that. Also, the tubes will need to be handled when open to add the buffer, but also to add the test strip. And so when we handle open tubes, there's always the risk of some spill. That means in that case, there would be a contamination of the surface, possibly of the gloves and so on, and also a risk of exposure of many of some previous injury or mucosa. Are there some other risk of aerosols? Normally, no. At least if the tubes are mixed when closed, in that case, there is no risk of other risk of aerosol. Also, since there is a risk of aerosol and or spill, there is also a risk arising from surface contamination. So we need to think about also the contamination practice. And in this case, there is no risk arising from waste handling because the tubes are eliminated in closed containers and incinerated on site. So looking at all that, we need to evaluate the likelihood of exposure. And in that case, I would say that exposure is possible. We cannot say it's not possible, it's really possible. And the likelihood will tend to be higher for HPV than for HCV and HIV. Why? Because of the natural prevalence of those different elements. Also, the likelihood of exposure is mostly through aerosol generation or a possible spill. Then what would be the severity of the consequences? Actually, in this case, we can consider that it would be low for HPV. Why? Because the people are vaccinated, but they could be moderate to high for the other agents, HCV and HIV, because people who would be exposed could develop infection and disease, and in some cases, there could be heavy consequences of the disease. So you see how we work. We collect the information, we identify all the situations of risk, and then we define the likelihood of exposure and the severity of the consequences of an exposure. And so that we can translate that into the risk assessment matrix, which is the upper part of this table. This table actually is in some templates that are supplied by WHO in the monograph on risk assessment. So in this case, you see in blue there that I put the different situations, that activities that could generate some risk. And for instance, there would be some risk of aerosol generation, and also from that aerosol generation, and that's mostly when opening the tubes. In that case, we can say that this initial risk is medium, and then and because of medium, and that risk would not be acceptable because of the possible consequences. Also, the risk from of a spill when handling the tubes, and also the risk from that spill, it's also a medium risk, and it's also not acceptable. And then there is also the risk of cut or injection. In this case, it's very low. Why? Because we don't use sharps, we don't use real glass, so there is the risk of that risk is very low. So we can consider that this is acceptable. And then the risk from handling waste is also very low because it's in closed containers, and it's something which is totally under control, I would say. So based on that, we can define that the initial risk, so the risk in the current situation, would be medium. So the initial risk is medium, and that means that we still need to do something because there are some risks which we don't think are acceptable, and because of that, that means that we cannot proceed to work without adding some more control measures. Okay? If doing the risk assessment, you end up with risk which is very low, in that case, you don't necessarily need to put control measure if you accept that it is acceptable. So you see, it's always important to define whether it is acceptable or not, and we will come back on that a little bit later. This is another way to use the same table. In this case, instead of putting the laboratory activities there, I put the different types of infection: hepatitis B, C, HIV, and the other infections. And in that case, we obtain something which is a little bit different. We still have risks which are not acceptable, and it's still a medium risk situation. And we also need to put. So I show you this other way to present things because you need to find your own way, and to find a way that where everybody agrees and which allows you to define what the level of risk, but also what is acceptable and what is not acceptable, because that will really kind of condition what we need to do afterward. So step number 3 is to develop the risk control strategy. So the needs there is mainly to reduce the risk associated with a possible exposure to aerosol when opening the tubes, or in case of a spill. Also to reduce the risk of spill and therefore surface decontamination. And then also the risk, we need to reduce the risk of contact of infectious droplets with non-intact skin, skin, or mucosa. So these are things we should really focus on. Looking at the context, the work will only start within 1 month, so we have some time to get prepared. And during that time, the people will be trained on the protocol. Another thing which is related to the context, I told you that the biosafety cabinet has not been checked and maintained, and it will not be possible to do that before 1 month, because we just don't have any funds to do that, and we cannot just plan for that because of that reason. Also, we know that there is no other biosafety cabinet that is available in the other labs and that we could use. So after our discussion, we decide that the risk control strategy is that even if the biosafety cabinet is probably not totally efficient, we will still use it. Why? Because it will allow us to reduce the risk of aerosol release. At least if there is an aerosol in the biosafety cabinet, it will be trapped at least partly by the. Of course, we would not take that decision if the biosafety cabinet was totally dysfunctioning and sending the air to the front window, for instance. But we'll do that when wearing some additional personal protective to reduce the risk of exposure of the personnel. Okay, so that's really the main idea that we have for the risk control strategy. What we also want to do is we want to speed up the funding for the checking, maintenance, and recertification of the biosafety cabinet. And so, this kind of decision is taken by the people who are in charge of management of the lab, I would say. After having decided about define that strategy, we need to select and implement risk control measures. So we'll really look at more specific things to do to really control the risk, to really reduce the level of risk. So for that, we will take additional precaution for specific steps for the dilution and mixing of the sample, and also to introduce the test strips into the tubes. For that, we'll use the biosafety cabinet, and as long as the biosafety cabinet has not been recertified, people will wear goggles or a face shield. You see illustration on the right hand side, and then we also wear 2 pairs of gloves. Why did we decide this? Because during work we could need to change gloves because they have been torn or contaminated, and in that case, when we wear 2 type of gloves, we can just take the outside one, and the skin still stays protected during that time. Also, as part of the risk control measures, we want to ensure that the current applicable work practices are followed properly. So we want to have not only have the staff trained by the manufacturer on this new testing, but we also remind a number of things, like that vortexing must be done with the tubes closed. Also, we want to remind the decontamination procedures and also the instructions for the elimination of waste. Another thing that we decided to put in place is that we need to check that all the staff, including possibly the future new staff, is vaccinated against hepatitis B. And so we could put as a condition that nobody that would not be vaccinated against hepatitis B could work in this project. So once we have selected the risk control measures, then we need to check a few other things. First, if those measures, if there is, they could be kind of compromised or affected by some regulations or some policies and so on. And that's the upper table of the table. You see that there is no regulation, there is no impact there. But that inside the institution, we currently have some SOPs, standard operating procedures, for the decontamination and waste destruction. It's a positive impact there. Those things are already there. And then we look at the different risk situations we have identified, and we kind of confirm the measures we'll take. So to control the risk from aerosols, we use the biosafety cabinet and also additional PPE. And so after doing that, we consider that the risk of aerosol is very much limited. And even if there would be an aerosol, the additional personal protective equipment, meaning the goggles or the face shield, would be enough to protect. And so we consider that the residual risk after application of those measures is very low. The risk from spill, again, is very low. It could be spills, but the consequence of the spills would be very low because of the use of the personal protective equipment and also because of the decontamination procedure. So in this case, there is still a risk of spill, but the residual risk and the risk of infection is also low. And of course, the 2 other risks were already very low, so that's not an issue. So is the risk acceptable after application of the measures? Yes, we think that the risk is acceptable. And so in that case, do we need to have additional measures? Probably not. And are the risk measures in play that we propose sustainable, effective, and so on? We think it's yes. So we go back to the risk assessment matrix to represent that, and in that case, we end up with some low level of risk, with a very unlikely exposure because of all the measures taken. So the residual risk is also low, and then we are satisfied. We can proceed with the work. You see that also in this template, and that's a template that is supplied by WHO. This has to be conducted by someone and approved by someone. So it's a kind of official document that will stay as a documentation. Still with respect to not the selection of the control measures, but the implementation of the control measures, we need to think about a number of very pragmatic aspects, like, for instance, communication and instruction to give to the staff, to the personnel who will work. As I said, we need to have them trained by the manufacturer, but we also need to prepare a specific instruction or SOP that includes new risk control measures. Also, the third bullet point there, and I think that this is very important, and I will explain why. We also need to inform the staff on the status of the biosafety cabinet. That we need to tell them that it might not be operated 100% efficiently because it has not been recertified, and also explain why we put those additional measures like goggles and face shield. Why do I think that it is important to give information about that? Because with a system like this, where each institution will evaluate their risk according to their way of doing, to their own situation and so on, you might end up with a way of doing which will be different from another lab. And so if people compare with a colleague that works in the other lab, they might be surprised and they might not trust what you are proposing. So in that case, you need to really be, I would say, transparent on the measures you will take in place. And it's also a way to have the people buying the proposal, the measures that you put in place. Then also for implementation, we need to think about the management of the implementation and the logistics. So for instance, we need to ensure that goggles and face shields will be purchased, that we have sufficient stocks of gloves because we'll use more. And we'll also need to remember that we need to obtain some budget and plan for the biosafety cabinet recertification. Okay, so collecting is one thing, and then we really need to think about the practical implementation of the risk control measure. Then we come to the last step, a review of the risk and the risk control measures. When finishing the first risk assessment, we need to establish the frequency of the review. So for instance, say, okay, we'll do this again next year. That's a kind of reasonable space between two risk assessment. Or we could also do it in case there is a change. And in this case, when the biosafety cabinet will be recertified, that will be a change. And so at that moment, we can reassess the situation on the basis of the result of the recertification. You see, the whole system is something which opened the door to different solutions. It's not just promoting one way of doing things. And the situations that you put in place, I mean, the measures that you put in place will really need to correspond to your own situation. And this is really what makes this system interesting, even if it looks more complicated to manage, perhaps. And of course, risk assessment this way, there are many more aspects that needs to be taken into consideration. So it might look more complicated, and this is why we will need some training. So Shazia, could you please summarize this part?
इस मरहले में हम एक केस स्टडी पे बात करेंगे जिसमें जो कि तेजी से खून की जांच के बारे में है। खून में मौजूद एजेंटों की तेजी से तशखीस करता है और ये तशखीस तैयार करंदा की हिदायत के मुताबिक की जाती है। पहला मरहला मालूमात इकट्ठी करना है। इस हिस्से में हम 3 किस्म के बारे में मालूमात इकट्ठी करेंगे। पहले हिस्से के अंदर हम खतरनाक हयातियाती एजेंटों की मौजूदगी के बारे में जो मौजूद हो सकते हैं उनके बारे में मालूमात इकट्ठा करेंगे, जैसे हेप बी, हेप सी, HIV और साइफिलिस वगैरह, कि ये आबादी में मकामी आबादी में इनका फैलाव कितना है, कितनी तेजी से फैलते हैं, वैक्सीन मौजूद है या नहीं है और इनका इलाज मुमकिन है या नहीं है। इसके बारे में तमाम की तमाम हम जांच को और मालूमात को इकट्ठा करेंगे। दूसरे मरहले में हम देखेंगे कि हम जो जांच का तरीकाकार इख्तियार कर रहे हैं वो कैसा है। जैसे हम देखते हैं कि सैंपल ट्यूब 2 मरतबा खोली गई हैं। इंक्यूबेशन और मिक्सिंग वर्टिसिंग जो है वो बंद ट्यूब्स के अंदर की गई है। इसके अलावा लेबोरेटरी और सामान कैसा है। लेबोरेटरी हम देखते हैं कि बुनियादी दर्जे की लेबोरेटरी का कमरा है जिसके अंदर एंटी रूम नहीं है, लेकिन वहाँ पर थोड़ी सी जगह है और बायोसेफ्टी कैबिनेट मौजूद है, लेकिन उसकी जांच और देखभाल की जरूरत है। सिंक मौजूद है, हेपा फिल्टर्स नहीं है, ऑटोक्लेव ऑटोक्लेव नहीं है, लेकिन फुजले को इंसिनरेटर तक भेजने की सहूलत मौजूद है। तीसरी जो जिसके बारे में मालूमात इकट्ठा करेंगे कि लैब का अमला और बायोसेफ्टी की सूरत हाल लैब में कैसी है। तो लैब का अमला हम देखते हैं कि बुनियादी तशखीशी मशकों में तरबियत याफ्ता और तजर्बाकार है। बायोसेफ्टी की जो PPE है वो बुनियादी नौइयत के मौजूद हैं, और लेकिन इस टेस्ट के ऊपर अमले को तरबियत भी देनी है और इसके साथ ही हेपेटाइटिस B की वैक्सीनेशन भी करवानी है। अब हमारा दूसरा मरला खतरे की तशखीस है। इस अमल में हम देखेंगे कि तमाम मुमकिन हालात जिनमें हम एरोसोल को पैदा होने का खतरे को महदूद कर सकें। हम देखते हैं कि वहाँ पर स्पिल जो है वो छोटी हो सकती है, सतह की आलूदगी का खतरा कम है क्योंकि आलूदगी से पाक करने का तरीका मौजूद है। तो नुमाइश का इम्कान मुमकिन है और नुमाइश की सूरत में नताइज जो खतरे का लेवल है वो कम से दरमियाने दर्जे तक का हो सकता है। जब हमने रिस्क को जांच की तो हमने देखा कि एरोसोल का खतरा दरमियाना दर्जे का है, स्पिल का खतरा दरमियाना दर्जे का है। हम यहाँ पर शार्प्स का इस्तेमाल नहीं करते हैं क्योंकि हम शीशे के बजाय प्लास्टिक का इस्तेमाल कर रहे हैं, तो शार्प से बीमारी का खतरा कम है और हमारे पास फुजला से निपटने का खतरा भी कम है। इस वजह से मजमूई तौर पर इब्तदाई खतरा जब हम मुंतखब करते हैं तो हमने उसको दरमियाने दर्जे का दर्जा मुंतखब किया है। इसके साथ हमने ये फैसला किया कि इजाफी कंट्रोल के इक़दामात के बगैर आगे काम करना हमें नहीं चाहिए। जब हमने ये बात का ये फैसला ले लिया कि हम इजाफी कंट्रोल के बगैर काम नहीं करेंगे, तो हमने इसको दूसरे तरीके से भी देख सकते हैं कि हेपेटाइटिस B की इन्फेक्शन का खतरा जो है वो कम है, हेपेटाइटिस C की इन्फेक्शन का खतरा दरमियाना है, HIV का खतरा कम है और दूसरी इन्फेक्शन का खतरा भी कम है। इसलिए हमने अपने रिस्क को दरमियाने दर्जे का कहा और हमने कहा कि हमें इजाफी इक़दामात लेने हैं। इजाफी इक़दामात अगर हमने लेने हैं तो हम सबसे पहले रिस्क कंट्रोल की हिकमत अमली तैयार करना है। रिस्क कंट्रोल की हिकमत अमली तैयार करते हुए हम देखेंगे कि हमने खतरा को कैसे कम करना है, कि एरोसोल की नुमाइश कैसे कम करनी है, SOP के ऊपर को कैसे बेहतर करना है, उस पर अमले की तरबियत के लिए हमें जो महीने का वक्त दिया गया है उसके लिए हमने कैसे तरबियत करनी है, और बायोसेफ्टी का इस्तेमाल क्योंकि हमारे लिए मजबूरी है, चाहे वो सर्टिफाइड नहीं है उसके बावजूद, तो हमने उसके ऊपर कौन सी ऐसी चीजें करेंगे कि हम इसको खतरे को कम कर सकें। चौथा मरहला हमारा रिस्क कंट्रोल की हिकमत अमली को मुंतखब करना और उस पर अमल करना है। उसके ऊपर हमने देखा कि हमने जो इक़दामात मुंतखब किए उनमें इजाफी एहतियाती तदाबीर हमने ली कि हमने टेस्ट स्टेप्स को मुतारफ करवा दिया, बायोसेफ्टी की रीसर्टिफिकेशन के लिए अपनी कोशिशें तेज कर दी और ये वाज़ेह कर दिया अपने अमले के कारकुनों पर कि जब तक वो रीसर्टिफिकेशन नहीं होती वो दो दस्तानों और हिफाजती आने का इस्तेमाल करेंगे बायोसेफ्टी में काम करते हुए, और सिर्फ बंद ट्यूब्स को इस्तेमाल किया जाएगा और फुजला को मुतादी फुजला के तौर के ऊपर ट्रीट करेंगे और अमले को HB की वैक्सीनेशन भी करवा देंगे। जब हमने ये तमाम चीज़ें की तो हमने देखा कि इसके साथ ही हमने ये देखा कि क्या कौमी या बैनुल-अक़वामी रेगुलेटरी अथॉरिटियों की तरह कोई तजवीज़ हमारे पास मौजूद है, तो हमने देखा वो हमारे पास मौजूद नहीं है, लेकिन हमारे अपने अदारे की हिदायत, पॉलिसियों और हिकमत अमली जो है वो हमें दी गई है, जिसके अंदर हमें बताया गया है कि आलूदगी को ख़त्म करने और ज़ाया करने से किस तरह से SOP फॉलो करनी है। इसके लिए हमने अपने खतरे को जिसके ऊपर इक़दामात किए वो ये था कि एरोसोल का खतरा हमने कम कर दिया कि हमने बायोसेफ्टी कैबिनेट को इस्तेमाल किया और उसके साथ हमने PPE को ज़्यादा कर दिया। स्पिल का खतरा हमने कम कर दिया कि हमने अपने PPE को ज़्यादा कर दिया और अपने खतरे के आलूदगी से निपटने के प्रोसीजर को ज़्यादा बेहतर कर लिया। कट चेन इन्फेक्शन का खतरा हमने कम ऐसे कर लिया कि हमने ग्लास के बजाय प्लास्टिक का इस्तेमाल कर दिया और फुजले का खतरा हमने कैसे कम किया कि हमने बंद ट्यूब्स का इस्तेमाल कर दिया। दिया। जब हमने इन तमाम चीज़ों को इस्तेमाल किया, तो जो हमारा खतरा, जिसको पहले हम मीडियम या दरमियाने दर्जे का खतरा हमने इब्तदाई खतरा अपना किया था, उसको हम अपनी तमाम इक़दामात को करते हुए, हिफाजती इक़दामात को मुंतखब करके उन पर अमल करते हुए हमने उसको कम दर्जे का, निचले दर्जे का खतरा बना दिया है। अब हमारे पास ये काबिल कबूल खतरा है। अब हमने क्या करना है कि ये जो हमने हिकमत अमली तैयार की है, इसको हमने अमली जामा पहनाना है, और उसके लिए हमने SOP में रिस्क कंट्रोल के तमाम इक़दामात डालने हैं, अमले की तरबियत करनी है और उनको बायोसेफ्टी की सर्टिफिकेशन तक वाज़े करना है कि हर सूरत वो हिफाजती ऐनक और दोहरे दस्ताने पहनकर अपने काम को मुकम्मल करें। बायोसेफ्टी की रीसर्टिफिकेशन की तरफ जल्दी जोर लगाना है। इन तमाम जब हालात को हम देखते हैं, तो अब हमने अपनी मुंतखब शुदा हिकमत अमली के खतरे के जो इक़दामात लिए, उनके हमने नाज़रसानी करनी है। जायज़ा लेते हुए हम देखेंगे, सबसे पहला जायज़ा हम उस वक्त लेंगे जब हम बायोसेफ्टी कैबिनेट की दोबारा रीसर्टिफिकेशन करेंगे। उसके अलावा हमें ये फैसला करना है जब भी हमारे काम करने के तरीके के अंदर कोई तब्दीली आती है या हमारे किसी इंस्ट्रूमेंट में कोई तब्दीली आती है, तो हम अपने इन इक़दामात को जायज़ा लेंगे, और इसके अलावा कम से कम साल में 1 मरतबा ज़रूर इन इक़दामात का जायज़ा लेना चाहिए। ओवर टू यू फिलिप।
Thank you, Shazia. Oops. So there are a few questions, but we'll keep that for session 4 because there are some interesting questions. So that's good. Finally, if we look at biological risk assessment, it's a way to define what are risk control measures that are adapted and sustainable to our situation. We've seen that it's a stepwise process. We also saw that it's a systematic process. It needs to be done with the same approach from one project to another, and also over time. If we repeat it in a few times, we should have comparable results because it's done the same way. It's also the basis for of the biorisk management system that you put in our organization. That means that it has quite a lot of impact on the functioning, the practical functioning of the organization. So who should do the risk assessment? Who should conduct the risk assessment? It's rather complex. It's technical. The technology, the methodology used itself is not so obvious. So we need to have people trained to apply this methodology. That means that you won't be able to do risk assessment like that right after this workshop. And it was not among the learner objectives of this workshop. This workshop, this webinar is just a first step, because you will need to be trained, and you will need to be trained in practicing the approach. Also, we need to cover a very wide range of information, not just biological agents, but lab activities, the facilities and equipment, how they work, and so on. Also the logistics. And so we need to involve the people who have the most knowledge in these different areas, on these different topics. That means that by definition, a risk assessment like that is a teamwork. It has to be done with the input of a number of different persons, of a number of different people with different experience and knowledge and so on. And then also the process is important for the organization, because it will impact the work process, but also the biorisk management, and also the way we manage projects and the future of projects and so on. So clearly there are responsibilities in doing that. It should be driven by a leader, someone who has a leadership position. It could be the principal investigator. It could be the biosafety officer. I prefer that it is the principal investigator, and the biosafety officer is more like a facilitator, challenging positions and so on. But anyways, those are different possibilities. And there is also a need for management approval. And so the best place for that is the institutional biosafety committee. If your institution has an institutional biosafety committee, in that case, that's really the place where such risk assessment should be reviewed and finally approved. If not, there must be some scientific committees, some management committees that should also do that, be able to do that. And if it's a very small structure, a very small organization, it could be the the head of the lab who takes that decision to approve the risk assessment. So to finish with this, there are quite a lot of information in the monograph on risk assessment. So I really invite you to read this with attention when you have time. You won't be able to do it after reading it, but it's very interesting to read. There are quite a lot of information there. Also, there are some other monographs of that biosafety manual that are also interesting for this, mainly the one on PPE and on biosafety cabinets, and the ones that deal with work practices. Also, in the monograph, the WHO provides a number of examples of application of key steps, and in that case, I put a few examples there, like routine smear preparation and microscopy, or large-scale culture of drug-resistant strain, and so on. Those are examples. They should not be copy-paste, but they need to be really customized to be adapted to your situation. But at least you will see the kind of things that are done and how it is done. Also, the WHO supplies 2 risk assessment templates, a short one, the one I used in this case study was a short one, and a long version. Those, I think, should be used as such first, so you could just use the forms that have been prepared by WHO. But I think that if you start doing it, in that case, in the future, you will adapt it and make it more suitable to your own needs. Also, WHO developed a few examples of fully completed risk assessment templates on Mycobacterium tuberculosis testing, on blood-borne pathogen diagnostic testing. This is the one I presented. On some influenza research work, and also on antimicrobial susceptibility, testing. Again, these are just examples. And of course, there are a number of things that that could be the same, like for instance about the agent, but all the things that are more local, more linked to your facilities, to your institution, to your working, need to be adapted. So it's really not the purpose of providing that is not so that it's copy pasted, but really to give you an example that you can do your own after that. So the next steps will be session 4, where I will answer your questions, the question you asked through the chat box. I propose that because I know that it's quite intense, a quite intense webinar. If you have more questions, please address them to the PEPSA office and they will send it to me. But please do that before end of the day on Friday. So that and then, as I said, I really hope that we will be able to so that you can work yourself on some case studies that are adapted to your own situations. So that's why my wish for the future. And now, please, Shazia, could you please help in translating?
हम देखते हैं कि हयातियाती रिस्क की तशख़ीस एक मुनज़्ज़म अमल है। रिस्क के कंट्रोल के इक़दामात के वाज़ेह और पायदार होने चाहिए। इनको मरहलावार 5 पहलुओं में तकसीम किया गया है और इनको उसी तरीके से हल करना चाहिए। मुख्तलिफ़ किस्म के ख़तरात के साथ मुख्तलिफ़ किस्म की सरगर्मियाँ होती हैं और इब्तदाई रिस्क की तशख़ीस का ज़रूर जायज़ा लेना चाहिए। और अदारे की रिस्क असेसमेंट कौन करेगा, इसके बारे में हम बात करेंगे। रिस्क ख़तरे की तशख़ीस के अमल की ज़िम्मेदारी एक पेचीदा तकनीकी और मुनज़्ज़म अमल है और इसके लिए तरबियत याफ्ता लोगों की ज़रूरत होती है। बहुत से मालूमात को कवर करने की ज़रूरत होती है, जैसे हयातियाती एजेंट, लैब की सरगर्मियाँ और सहूलियात वगैरह। इसके लिए इंतहाई इल्म वाले बंदे को मुनज़्ज़म करना चाहिए। ये एक टीम वर्क है और इसके काम करने के अमल और रिस्क मैनेजमेंट के लिए बेहतर है कि आपकी सीनियर मैनेजमेंट इसको लीड करे और ये बायोसेफ्टी की कमेटी या इंतज़ामी कमेटी उसको बाकायदा अनाउंस करती है। WHO ने जो मिसालें दी हैं, रिस्क असेसमेंट का एक मुकम्मल मोनोग्राफ है जो कि बेहतरीन तरीके से आपको पढ़ना चाहिए, बहुत अच्छी मालूमात फराहम करता है। इस मोनोग्राफ में रिस्क की कंट्रोल के मुख्तलिफ तरीके बताए गए हैं जो कि मुख्तलिफ मिसालें देकर वाज़े किया गया है। ये मोनोग्राफ में दो किस्म की टेम्पलेट्स बताई गई हैं, एक छोटी मुख्तसर टेम्पलेट है और एक लंबी टेम्पलेट है, जिनके अंदर मुख्तलिफ मिसालों के जरिए इसकी तशखीस और इक़दाम को बताया गया है। बेहतर ये है कि अगर इन चीज़ों को आप कॉपी पेस्ट मत करें और अपने अदारे के ज़रूरियात के मुताबिक अगर आप उसको अमल करने की कोशिश करेंगे तो आपको बेहतर नतीजे मिल सकेंगे। हमारा अगला सेशन 23 जून को है, जो कि सवालात और जवाबात पर मबनी होगा। अगर आपके कुछ सवालात हैं तो कोशिश करें कि उनको जुमे से पहले तक आप हमें पेप्सो ऑफिस में ईमेल कर दें, ताकि हम अपने अगले सेशन के लिए उनके बेहतर जवाबात दे सकें। उम्मीद करते हैं कि इस वर्कशॉप के बाद आप अपने अदारे के अंदर कोई प्रैक्टिकल वर्कशॉप्स भी करवाएंगे। बहुत शुक्रिया।
Thank you so much, Dr. Philippe and Dr. Shazia. Evaluation link मैंने chat box के अंदर डाल दी है, प्लीज उसको फिल करें। जो लोग evaluation फिल करेंगे उन्हीं को सिर्फ क्विज मिलेगा। evaluation लिंक आज रात 12 बजे तक ओपन होएगी, उसके बाद वो क्लोज हो जाएगी। अगर आपने उस टाइम तक नहीं फिल की, आप बाद में हमसे आके क्विज मांगेंगे तो हमें आपको अकोमोडेट करना मुश्किल हो जाएगा। तो प्लीज evaluation लिंक मैंने चैट बॉक्स के अंदर डाल दी है। इट्स गोइंग टू स्टे देयर फॉर द नेक्स्ट 5 मिनट्स बिफोर वी क्लोज द सेशन। आप बेशक लिंक को अभी खोल दें, बाद में फिल कर दें। इट विल बी ओपन टिल मिडनाइट टुनाइट, लेकिन ये लिंक आपको बाद में ईमेल नहीं होगी। और अगर आपको क्विज नहीं मिलेंगे, बाद में आपको क्विज नहीं मिल सकेंगे, सिर्फ उन्हीं लोगों को मिलेंगे जो ये लिंक फिल आउट करें। Other than that, thank you so much for attending. अगर आपके कोई questions हैं, मैंने अपना email address भी chat box के अंदर डाल दिया। अगर आप लोगों के पास नहीं था, please send us any questions that you have, and the next session is just going to be question and answers. And thank you so much. Okay, everyone, I will be closing the session now. मैं इस सेशन बंद करने वाली हूँ. एक बार और evaluation लिंक चैट बॉक्स के अंदर है, प्लीज इसको अभी ओपन कर दें. बेशक आप इसको बाद में फिल करें, 12 बजे तक की लिंक ओपन होगी, लेकिन प्लीज इस लिंक को कहीं सेव कर दें क्योंकि we will be closing the session now, तो फिर ये लिंक नहीं दोबारा सेंड होगी. एंड जो लोग क्वेश्चंस पूछ रहे हैं अबाउट द फर्स्ट क्विज, मुझे 5-6 क्वेश्चंस आ चुके हैं, उसकी लिंक सबको सेंड कर दी गई थी आफ्टर द फर्स्ट क्विज. जिन लोगों ने उसकी एवेल्युएशन फिल आउट किए थे उनको फिर क्विज भी सेंड कर दिए गए थे. तो अगर आपको नहीं मिली, इसका मतलब है कि आपसे शायद वो ईमेल मिस हो गई है. अब वो सेशन क्लोज हो चुका है, वी विल नॉट बी एबल टू सेंड इट टू यू अगेन. लेकिन अगर आपको अभी भी कोई वैलिड रीजन थे या कुछ, आप मुझे प्लीज ईमेल कर दें, हम आपकी रिक्वेस्ट फॉरवर्ड कर देंगे आगे. एंड विद दैट मैं लास्ट बार लिंक शेयर कर रही हूँ एवेल्युएशन की, प्लीज इसे आप खोल दें क्योंकि इसके बाद वो सेशन क्लोज हो रहा है एंड द लिंक विल नॉट बी अवेलेबल चैट बॉक्स के अंदर. थैंक यू एवरीवन फॉर अटेंडिंग. अल्लाह हाफिज.